Namaste to all. Welcome to the next session on drug designing. So in today's session, we are going to discuss about the drug target, mainly about the DNA specific inhibitors of transcription. See, the main idea yesterday we have, that is in the previous session, we have already discussed about the drug targets, what are the different types of drug targets, how they play an important role for binding, as well as how they are actually stopping the particular reaction or increasing the particular reaction. So in this session, what we are going to see here is there are so many organisms are there, or you can like prokaryotic organisms, like infection causing organisms, bacteria, in which there you know that during the transcription process, there are various steps which occurs, like RNA polymerase, they will search and they will bind at a particular location like promoter or operator, and they will start to function. So in the same manner, if we inhibit the process of the binding of the RNA polymerase or the complex formation of the initiation complex formation, so what will happen here is we can inhibit the process of transcription, thereby the protein which has to be produced by the bacteria, which leads for the infection, like toxic material or any enzyme which causes the virulence to be increased by the bacteria causing the infection, that will be decreased. The other method by which in this we are doing is making a mimic of the DNA itself in some cases. So what happens is when they make a mimic of the same kind of nucleotide, these molecules, when they bind with them, then what happens is they will inhibit the process of this particular organism, and thereby they can stop the reaction. So this particular method of epigenetic level, or you can say like a chromatin, the euchromatin level of inhibition, which leads for the transcription to be inhibited in prokaryotes, and in some cases of the eukaryotes, is actually the process of the DNA specific inhibitors of transcription. So we will see this in detail right now. So the first aspect in this is we have to know about the central dogma of molecular biology. So we know that from the DNA, by the method of transcription, we are getting our mRNA, and by the process of translation, we are having our protein. So the transcription is the process by which the DNA information is copied to an mRNA molecule, and this is further used as a template for translation and the process by which the proteins are synthesized. So this is, in simple way, we can say this as our central dogma of molecular biology or expression, gene expression in all the cases. So the inhibition of the transcription can be done by various methods. The first thing is targeting the PIC, that is a promoter initiation complex. So you know that in the promoter initiation complex, what is happening? There is an assembly which takes place by the transcription factors, or with the help of the RNA polymerase and sigma factors and other factors, they join together to initiate the particular process. So if we target this promoter initiation complex, we can inhibit the process of transcription. The second process in this is reducing the DNA accessibility to transcriptional machinery through epigenetic modifications. So what we are doing to reach to the particular level in the epigenetics, that is in the euchromatin level, if we reduce the DNA accessibility, that is to find out that this is a DNA in which it is taking place, so that activity, if we stop by some of the enzymatic action or by some of the drug method, then what happens is the transcription can be inhibited. The third method in this is slowing down the RNA elongation process. So what happens once the RNA elongation is being stopped, or only the initiation happened, not the elongation, then what will happen? It will not be read completely, and only the small stretch will be only read, and the small amount of protein will be only released. Thereby, it is also, or you can say, a small amount of the RNA only will be synthesized. So what happens? We will not have, or the particular organism will not have the mechanism to go further, and they will stop this process itself. The fourth method in this is covalent or non-covalent modification of RNA polymerases. So you know that there are so many the subunits are there. So if we modify any particular subunit, then what will happen is it will stop the particular reaction and the complex will not be formed to form as an active RNA polymerase. So that process of stopping by the modification process only. So let's see this in detail with the help of examples and the particular drugs what we can use here. So the first example here, what we can take here is rifampicin. So rifampicin is also like a penicillin category only. So rifampicin, if you see, binds with the beta subunit of prokaryotic RNA polymerase. You know that in RNA polymerase of prokaryotes, we are having alpha, beta, and beta prime. Beta plays a very vital role. What it does, it will actually identify the location and elongate on the upstream position. So what happens near to the promoter, it will just join and it will start to elongate themselves. Then only the process of the transcription can be started up or initiated. This open complex formation, or from the closed complex formation to the open complex formation, this, if the rifampicin is there, it will block that and the process of transcription will not takes place. So it is an inhibitor of the prokaryotic transcription initiation, and it binds only to the bacterial RNA polymerases and not to the eukaryotic RNA polymerases. Therefore, rifampicin is a powerful drug for treatment of bacterial infections. So in which bacteria we can use? The highest level in which rifampicin is used is for the treatment of tuberculosis and leprosy. You know that tuberculosis, it is a very deadly disease. So what happens is in the lungs, it is getting affected, and leprosy on the skin, epidermal level. So what happens is to treat them with the normal antibiotics, it is difficult. So rifampicin is the source by which the transcription can be inhibited, and thereby the process by which the bacteria is replicating, or you can say like producing, the translating the proteins, that process will not take place, and thereby the process will be stopped, and thereby the the treatment will be successful. So this has to be taken by the patient for a nearly like 1 year course. So in a different terms, they have to take it. So rifampicin is the strongest drug for the treatment of the tuberculosis and leprosy. So this is the modeling by which we can understand that how the structure, this is the structure. When we are doing the 3D modeling of this particular structure, we can know that these are where they are getting targeted. So once the targeting is being done on the beta subunit of the RNA polymerases, then what happens is the function of this, that is the binding of the polymerase becoming as an active molecule, will not take place here. The second in this category is actinomycin D. So actinomycin, from the actinomycetes, basically from that they have extracted this. So the actinomycin D, if you see, this antibiotic inhibit the transcription in bacteria as well as in eukaryotes. So if you see how it is taking place, the planar position of this molecule inserts like an intercalates into the double helical DNA between the successive GC region or GC pair region, deforming the DNA. So what happens when they bind inside in between like GC rich region? So what happens if the structure is like this? They go in a deformed form. So what happens? The deforming form of the DNA will not allow for the transcription process now. So inhibits the RNA elongation by restricting RNA polymerases along the template. So now, since the deformation is there, so what happens? The RNA polymerase cannot continue further. So the elongation will not take place, and thereby the process is stopped. And it is basically on the concentration dependent action. Concentration dependent action means how much the amount of actinomycin D has been provided. Based on that only the reaction can take place. Then acridin is also functions in a similar fashion. So acridin, you know that ethidium bromide or acridin and all, they are intercalating agents. So in the same manner, this actinomycin D also works. So this is the structure of actinomycin D. This whole structure is actinomycin D. and this is our acridin. So if you see the structure, almost it is similar, but only thing is the side chains, if you see, this side chains are not present. So what happens is this almost looks similar to, you can say, like a pyrimidine structure, almost like a purine structures. So the purine structures, when they are seen, or nucleotide like adenine or guanine structures, if you see, in that part, the guanine actually plays a very vital role, because it is also having the NH2 and O is there. So this can pair up there. Okay, so when they intercalate in them, so what happens? They will form a deforming DNA. So that is the important part of the actinomycin D. Next in this category is our alpha amanitin. So this is a cyclic peptide of 8 amino acids. So you can see here there is a cyclic peptide, it is of 8 amino acids in this. So 8 amino acids are there in this. So this is a cyclic peptide. So it is a defense mechanism by Amanita phalloids. Okay, phalloids. So Amanita phalloids, this is an organism in which this is a defense mechanism by which they are using the alpha amanitin. But now what is happening when we are extracting this alpha amanitin and using it, this acts as a selective inhibitor of polymerase 2 and polymerase 3. So I think you must be knowing what is polymerase 2 and polymerase 3. In eukaryotes, we have different types of polymerases like polymerase 1, polymerase 2, polymerase 3, polymerase 4. So like this, different types are there for RNA synthesis, for tRNA synthesis, for mRNA synthesis, and for some of the snRNA synthesis and other things. So in the eukaryotes, in some places, if we are using the alpha amanitin, then what happens? They are selective inhibitor of RNA polymerase 2 and RNA polymerase 3. So this alpha amanitin, they interact with the bridge helix of polymerase 2. So this interaction, what they do is, bridge helix means interacts with the bridge helix means almost in the polymerase 2, the structure wise. Okay, if you see the structure wise, it's little bit different. Okay. So in that, the bridge structure they form, so then only they can be active. So that structure is being broken down, or you can say like it is being deformed almost. So this interaction interferes with the translocation of RNA and DNA needed to the empty the site for the next round of RNA synthesis. So what happened? It will take a delay. So from one position to next position, when they have to go, it will not go properly. So what happened? Once they are not going properly, the RNA which has to be synthesized, it will not take place properly there. So no effect on the affinity for NTPs and the phosphodiester bond can be formed here. So no much difference is there with that, but what happens? It delays the process because of the translocation which is taking place here. So non-competitive binding slows down the rate of elongation. So since it is taking a long time for movement, so that what happens is it will not move forward. So the elongation process is delayed here. So since it is getting delayed, what happens? The enzyme or the toxin which has to be produced, that will not take place, or the protein metabolic regions, what are the things is required for metabolism and all, that will not happen. So due to these reasons only, they are saying that there is a slowdown of rate of elongation. So alpha amanitin can also be used to determine which type of RNA polymerase are present. Okay, so that is also used here for the determination purpose, and this is done by testing the sensitivity of the polymerase in the presence of alpha amanitin. So the testing process, if you see how it is done, is based on the sensitivity aspects. So here, since the sensitivity for the polymerases which is there is actually very low, because why we are saying the sensitivity is high or low is because once it is getting delayed, okay, once it is getting delayed based on the process of the elongation only, we are saying that how the sensitivity is working. So if you see the polymerase 1 is insensitive. So what happened? There is no binding at all. Okay, because RNA, you know that RNA polymerase 2 is usually required for mRNA synthesis. So in the mRNA synthesis only it is going to affect. It is not going to affect any tRNA synthesis or rRNA synthesis or snRNA synthesis. So if you see in polymerase 1, it is insensitive. So no reaction is going to take place for the RNA polymerase 1. But in RNA polymerase 2, if you see, it is highly sensitive. Highly sensitive means inhibited at 1 microgram per ml level. So what happened? In 1 microgram level, if the DNA is getting delayed or it is getting stopped, then what happens is the molecule which has to be formed as RNA will not take place. So that is why the mRNA process here is getting hindered or stopped here. The RNA polymerase 3, if you see, it is moderately sensitive. Moderately sensitive means it will also stop, but inhibition rate for that is almost like 10 microgram per ml. So in that 1 microgram is there, means one position like that, it is getting affected every each minute or for ml. But if you are 10 microgram means it is little bit moderately sensitive it is. And in the same manner, the RNA polymerase 4, which is there, it is slightly sensitive. Slightly sensitive means inhibited at 50 microgram, little bit more. Okay, it requires little bit when compared to the polymerase 2, it is little bit low only, but the amount in which it is taking place is like 50 microgram per ml. So it is not much. Okay, slightly sensitive it is. So the use of antibody drug conjugates. So in some cases we have seen that the antibody and the drug, both the things has to be given simultaneously, like immunoglobulins along with the drug has to be given. So then only it will work. So in those cases, what is happening is this amanitin which is there. So how it works is, this is a water soluble structure, low aggregation even at higher dose in vivo condition. So low aggregation means joining is very low here. So it is a water soluble structure. The second thing is very effective against the multi-drug resistant cells. So multi-drug resistant cells means in cases like organisms like you can say tuberculosis or related to staphylococcus, or you can say like cancerous cells also some cases, or some cells in the body also will becomes like a multi-drug resistant. So in those places it is very effective. The inhibition of amanitin induces apoptosis in targeted cells through antibody. So what happens where all this alpha amanitin is present in the targeted area? So what happens is they induce the apoptosis, and thereby what happens is the natural apoptosis process also starts there, and it will inhibit the particular infected cell. So that is the important part here. The another thing is studies in the mouse model for prostate cancer have been highly successful. So they have found that the rate of the prostate cancer formation is actually decreasing, and thereby it actually helps for preventing the prostate cancer. So this is the process of how the RNA polymerase activity in presence of alpha amanitin is working. So if you see the green color, the top one, green color, this is actually about our RNA polymerase 1 actually. So in the RNA polymerase 1, it is insensitive, we have said. Na? So in this, you can see like 100 level also, it is actually very straight only. There is no difference, no lowering down, nothing like that. But in the concentration of 1 microgram per ml concentration, if you see, it is actually decreasing itself. In the first level itself, it is actually decreasing. In the third level, that is polymerase 3, if you see, it is almost equivalent to almost like 10 microgram level, or you can say like 20 microgram level. In that level, it's actually decreasing. And in the further, we have seen that the 4 is almost like 50 microgram level. So it is further far. So the time taken, if from this diagram, what you can, or in the chart, what we can understand is the time taken in which, or the maximum activity in which it is taking place is, it is in the polymerase 2, in which the mRNA will not be synthesized by the transcription process. So the next process in this inhibitor is the endogenous inhibitors. So if you see the endogenous inhibitor, endo means inside, inhibitor means it is stopping the reaction. So what is taking place? The methylation pattern of CpG, okay, CpG means like cytosine and guanine. So islands are there in the upstream of promoter. So the Cp stands for promoter, C is for cytosine and guanine. So here what is happening is, you know that in the promoter region, upstream of the promoter region, we are having a GC rich region. So in this GC rich region, when the methylation takes place, they will start to either they will bind back or they will open up. So, or loosen up, you can say. So in this particular scenario, okay, so the normal CPG islands other than those near the promoters are methylated, and those near the promoters are unmethylated. That is, little bit far away they are methylated, near the promoter they are unmethylated. So what happens, if it is methylated, they will start to open up. If it is not like that, they will be very close. Okay, all the things will be close. So from this diagram, you can understand that how the process is taking place. So if it is methylated, okay, so if it is methylated, all the things will be nearby. Okay, if it is hypomethylated, means more amount of methylation is taking place in the case of like cancer cells, then what happen, everything will separate. Okay, they will open up, everything will be separate. So what happened, the transcription will be taking place, translation will be happening, the excited amount of replication is also taking place. So many things are happening. So that is why the tumor, this is actually like a cancer tumor suppressor genes and oncogenes. But once they are hypomethylated, this is being expressed instead of suppression of the tumor genes, they will express themselves as oncogenes or cancerous genes. So what happens, the cancer will be showing off. So that is the reason. Okay, so in this process, if you see, the deacetylation of lysine residues of N-terminal histone octamers will take place, and the accessibility of the genes in euchromatin and those in heterochromatin are reversed. So that is the process here. So you can see in this process, the first step you can see here, there is a methylated repetitive sequence. So the repetitive sequences are there, they are nearly close. So if you see the hypomethylation of the DNA, you can see everything is going far away, or some of the molecules are actually being exposed, you can say. In the other case, you can see that normal cell, that is the histone modification is taking place. So what is happening when the histone modification takes place? They are nearby, very close. The octamers are very nearby. They will not allow the DNA to get loosened up for transcription or translation. Once they are actually losing the property of histone methylation in the cancerous cells, then what happened? They are very loose. So what happened? More transcription will take place repeatedly. So that is why the cancerous cells will replicate and grow again and again. The third step, if you see here, that is the unmethylated CPG islands and methylated gene bodies are there. So this is the promoter regions. Okay, these are all unmethylated. These regions, if you see gene bodies, they are all that is the methylated. So what is happening? They are all there. So there is no expression which is taking place. Why? Because we know that when it is methylated, they are all close by. So what happened? The transcription will not take place. But during the unmethylated case, they are all far away. So what happened? The process can start, but it has to bind. The polymerase has to bind at the promoter region. So what happens now? So in this the hypermethylation case, what is happening? Hypermethylation, you can see that the genes are also expressed. The promoter region is also present. So what happened? They start to express themselves. So what happened? The transcription of exon 1, exon 2, exon 3, everything will start. Okay. Then the last stage, if you see here, the histone modification, that is the open chromatin configuration, and the same thing only, almost you can say the same thing only. So acetylation is taking place here. So that is also making like far away, but during the loss of the histone acetylation, they will be very close. So expression will not take place. So what is the meaning of this acetylation? That is, those genes which has to be stopped will those genes which are actually stopping this cancerous mechanism during the normal process will not occur during the cancerous process because of the loss of histone acetylation. So both the things, both the process are taking place. In one, expression will not take place. In another case, the expression will take place. So again and again, some molecules will be produced, and those molecules which inhibit this will not be produced in the cancerous condition. So that is what the endogenous inhibitors are. Okay. So the next is the reverse transcriptase. So if you see the reverse transcriptase, we have already seen in the organisms like viruses that they use the enzyme called as reverse transcriptase to generate the complementary DNA, that is the cDNA from the RNA template. And this process is called as reverse transcription. HIV, when it comes inside, HIV infection when it comes inside, for the analog of the TTP, that is the thiamine triphosphate, which is there, for that what we are doing is TTP. So what we are doing is we are giving AZT, that is 3' azido 2' 3' dideoxythymidine. When we give this, what is happening? The AZT is taken up by susceptible T lymphocytes. So those which are very weak, that will be taking this AZT. So this will be present. So what happens? Viruses, we know that they will stop the mechanism of T lymphocytes. So once they are inside the AZT, what happens? The reverse transcriptase, when the organism releases the reverse transcriptase, this has more affinity towards the AZT. The reverse transcriptase has more affinity towards the AZT, that is as AZT triphosphate, than the DTTP. So what happened? Unlike the mammalian polymerases, they will just bind with this. Once they are binding with this, what they do, since they are lacking the 3' OH, they cannot initiate the process. So what happened? The viral replication will be terminated. So by this mechanism, in nowadays, the HIV infection patient who are there, they are giving this AZT to prevent the replication and to increase the level of the HIV in their body. Already affected things cannot be retrieved, but those things which can be prevented is being done with the help of the AZT. So if you see the structure, this is the structure of the AZT which is present here, and the mechanism, as I said, so what is happening? The reverse transcriptase mechanism is being stopped here by this AZT. So what happens? So there is no replication which is taking place. So these are the different references which I have taken this for this particular session. So if you want, you can refer this for your future. Okay. So I hope that you got an idea about how the inhibition of transcription is taking place by different drug sources and by the enzymes. And thank you and have a good day.